All right. Well, why don't we go ahead and kick things off? For those of you who do not know me, my name is Ryan Beebe, and I'm a professor in the biology department here at Calvin University. And I'm actually the vice president of the West Michigan chapter of the ASA. And I'm going to be serving as the MC for the event tonight. And I'd love to begin with all of you joining me by bowing your heads so we can have a word of prayer together. So please join me. Father God, we thank you for this day, and we thank you for the beautiful day that many of us were able to enjoy a little bit here in West Michigan. And we thank you for the opportunity to be able to gather together here tonight. It's no secret that we've been at the end of a difficult time, and we're still in the midst of a major struggle in our world dealing with this virus. And we thank you for the opportunity to learn a bit tonight about something that I think gives a lot of us hope as we move forward into these coming months, that we're hearing about these vaccines being close to deployment and hopefully getting us to the end of what has been a very difficult chapter. And we thank you for Anding's willingness tonight to share a little bit of her expertise with us to help us understand a little bit more about vaccines and how they work and how they're developed, and whatever other words she has to share with us tonight, Lord. We pray that you can bless the preparation that she has done and give her clarity of thought and clarity of speech to be able to impart what you would have her share with us here tonight. Pray that you can bless everyone who is joining us here within this group and bless our conversation and fellowship together as well. In your name we pray all these things. Amen.
Amen.
Well, I'd like to give a welcome to all of you who are joining us on here tonight, certainly to the folks who are members of the West Michigan chapter, but also if we have any folks joining us from the Wheaton chapter that we send an invitation out to as well, special welcome to all of you. And a special welcome to the Calvin community. I know I see some colleagues on with us tonight. I know I saw at least one or two students on here as well, so welcome to all of you. And a special welcome to anyone who has never been to an American Scientific Affiliation event before. While our West Michigan chapter is kind of the new kid on the block a little bit right now, the ASA is definitely not. Just a little bit about the organization. It was founded in 1941 as an international network of Christians in science. And even today, the organization continues to work towards integration of solid science with theology to help serve both the church and the scientific community as well. And I know that the organization really seeks to create an environment for dialogue and discussion that is marked by courtesy and respect, even when we might disagree with one another about the different ideas or topics under discussion. If you're not familiar with the ASA at all, you can learn a lot more on the website. So it's asa3.org. You can learn about the organization. You can see bits about the journal, Perspectives on Science and Christian Faith, which is the flagship journal for the organization. You can find out about how to become a member as well. I'd love to point out for all the students that there is actually a free student membership option where you can join as a non-voting member and get free electronic copies of the quarterly journal. For just $20, you can be a full student member with voting rights and get paper copies of the journal as well. So really, really great opportunity for the students who are interested in careers in science to be able to join what is really an outstanding organization and be able to network with individuals from a whole bunch of different scientific disciplines, all of whom are Christians as well. So a really, really cool opportunity. We will certainly have time after Dr. Shen's presentation for Q&A and for a discussion and just some fellowship with one another as well. You can hang on to your questions until the end of the conversation, till the end of the presentation if you would like, and you can ask them orally at that time. You can also submit questions in the chat as well, and we will sort of look through those after the presentation and pull out questions there as well for Dr. Shen. And we'll also, again, have some time for fellowship for folks that would like to stick around after the Q&A as well. But without further ado, I will take a moment just to introduce my colleague Anding, and then I'll turn it over to her. As you might have surmised, Anding is one of my colleagues here in the biology department at Calvin University. She is not a West Michigan native. She grew up in Shanghai, China, And she came to the U.S. as a foreign exchange student during her final year of high school. She went to high school then in Washington, D.C., at the very same school where a young Chelsea Clinton was actually taking some classes at the time. Anding tells me Chelsea wasn't in high school with her, but was like a junior high student taking some high school classes while she was there. So that's a fun fact. And from DC, she then went to Philadelphia and attended Drexel University there, where she studied biological sciences. And after completing her bachelor's degree, she moved on to Johns Hopkins University in Baltimore, where she completed a PhD in molecular medicine and had the great Francis Collins as her commencement speaker and commencement singer, I guess, as well as he is known for. She completed a postdoctoral fellowship at Johns Hopkins before coming to Calvin, and she joined the faculty at Calvin in the fall of 2005. In our department, she teaches courses in cell biology, microbiology, and immunology as well. And she directs a very active research program that focuses on HIV latency in T cells from your immune system. The two of us actually met at an ASA event. So it was the summer of 2006. I was a recent Calvin biology alum. She was a new Calvin biology professor. And we were attending the 2006 meeting of the ASA that was hosted at Calvin. And we were on a bus together taking a field trip out to Lake Michigan to one of the parks out there. And that's where we first had some great conversations. And I've come to know her since I've been back at Calvin on the faculty as just a wonderful colleague and a really outstanding research mentor to our students. And I know that I can speak for all the leadership in the West Michigan chapter that we are very grateful for her willingness at the end of what has been a very busy semester to share a little bit of her expertise with us tonight. So Anding, I will yield the floor to you. And again, folks can type questions into the chat box or just save them until the end of the presentation. Anding, please.
All right. Yeah, thank you, Ryan, for that introduction. And I see some friends here as well. So it'll be my pleasure to share what I know about this topic with you guys. And I hope to leave a significant amount of time at the end so we can have some conversation or dialogue. So let me start sharing my slides. Okay. Can you actually see my first slide? Okay. All right. So we're going to be talking about vaccines, and particularly in the context of our pandemic. So as I think all of you know, this disease started in late January and February. And when we hear about this, it came out of China. And then, of course, this is my home country. So I paid a very close attention to it. Started out in Wuhan, and then it spread quickly all through China. And so this is so I took a screenshot from the Johns Hopkins dashboard. And you can see by then it was mainly just in China and some neighboring countries. And then within a month's time, it spread all over the globe. You can see all those dots everywhere. And then by late March, U.S. surpassed China to become the number one. And as all of you know, we still hold that position today. Here is today. I took a shot today. And then the number of cases we have right now in the world is more than 100 times than the cases back in March. So this pandemic spreads really quickly, and we have a hard time really containing it. So if you look at, can you see my mouse here, cursor here? Yeah, so this little bump was back in March, April. And then global lockdown really slowed down the pandemic for a while. And then we had another peak in the summer. And then now we're in a dramatic peak right now in the fall as the Northern Hemisphere going into winter. So we're really having a hard time with this pandemic. And all of us, our lives have been changed because of this. So I took this picture while I was getting tested for COVID before we returned in-person instructions at Calvin. And then I took this picture on the very first day of classes. And everywhere we went, everywhere we go, we see that we're asked to wear masks to keep social distancing. And then that kind of becomes the norm in our lives. So this is a picture I took for my lab class. This is immunology class that I taught this semester. And you can see all students are wearing masks, face shields, and gown up. And then this is another interesting site. This one night I was just walking around the campus and I saw the orchestra was practicing under a tent outside. And everybody is wearing masks, including the wind instrumentalists. I don't know how they do it. So it was, life really has changed. So this is another picture I took this fall when we had, this is a Chinese church that I attend. I also attend Madison Square. And then we actually moved outside. When the weather is good, we're meeting outside. For the rest of the time, we're actually meeting on Zoom. So I'm pretty sure this is familiar to you as well. So we're definitely in the era of pandemic. This pandemic is caused by a virus called SARS-CoV-2. So it's SARS coronavirus number 2. Number 1 caused SARS about 17 years ago. This is the RNA virus, and it has the way of transmission is through droplets, aerosol, and a surface. So this is very difficult for people to avoid. So essentially it's airborne. It also has a quite high transmission rates, at least twice as more transmissible than flu. The variant, the mutant that actually is right now all over the world did not start in China. It actually started in Italy back in late February and early March. And that particular mutant is a lot more transmissible. And the virus causes a pretty high fatality rates. Earlier on, the case fatality rates are especially high. Right now, it has actually dropped over time, partly because we know how to treat it a little better. Partly is also the hospitals are not as overwhelmed. However, if we don't really control it, the healthcare system gets overwhelmed, then we might actually see an increase in fatality rates. And it's especially bad in people who are elderly or have underlying issues. There is no magic treatment. In other words, there is no treatment that can actually absolutely prevent people from dying. So this is another reason why it's very difficult to handle this pandemic. So to slow down the pandemic, to prevent death, prevention is key. There are 2 ways to prevent, essentially. One is avoid contact between people, and the other is generate immunity. So let's look at it, how to avoid contact. So I borrowed this slide from a presentation at a COVID conference I attended, virtual conference in July. It's organized by the International AIDS Society. And so my field is in HIV. So, and in the HIV field, scientists are actually taking on the mantle of organizing conferences and organizing scientists to deal with this pandemic. Before COVID, the the pandemic or any pandemic that has this widespread, extensive spread is actually HIV and AIDS. And it's still ongoing, although we do have treatment for HIV, so we don't hear about it that often anymore. However, if I'm allowed to digress a little bit, we still do not have a vaccine for HIV yet. This is after 40 years. So back to avoid contact. So what are the ways we're doing? So you're probably all familiar with this. Social distancing, hand washing, and wearing masks. More and more data are showing that wearing masks is the most effective way of preventing COVID transmission. And there are various other ways public health officials are doing to really slow down the pandemic, including some form of lockdown. Now, in terms of generating immunity, that's another great way to really control the pandemic. Before we go there, I want to talk about the immune system a little bit. The immune system really is an amazing system. God gave us this system to help us to defend against pathogens, invaders. The immune system has two arms. One is called innate immunity. These are cells, various types of cells that can recognize pathogens by some kind of pattern. They're unique to pathogens, and then humans or whatever the host doesn't have. And they can help to kill or remove pathogens accordingly. Another arm of the immune system is called adaptive immunity. And this arm can recognize specific antigens associated with specific pathogen. So this is, in other words, this arm of the immunity can actually look at a pathogen and knowing for that particular pathogen, we have particular weapons to deal with it. So if I want to use the analogy, the innate immunity is kind of like when you take vitamin C or zinc or herbal supplement when you feeling like you're getting sick, whereas the adaptive immunity is like taking Tamiflu, a particular very specific thing for a very specific pathogen. Now, the way it works in the adaptive is that it's through a mechanism called clonal selection. So we have B cells and T cells in the billions in our body, and each one of them are actually carries a specific receptor. And they're specific for particular antigens or pathogens. So when a particular pathogen comes in, then one particular or several hundred particular B cells or T cells get selected. They are the ones for that particular pathogen. Once they are selected, then they can clonally expand to make a clone army of themselves. And that's why it has a powerful army of cells once the adaptive immune system is selected, is activated. Now, it actually takes time for the selection process, and that's why the adaptive usually is slow the first time around, the first time when the pathogen enters the body. Once they're activated, then they can actually do their jobs. So the T cells are good at killing infected cells, and the B cells are good at producing a very powerful molecule called antibodies. And antibodies can actually block the pathogen and get rid of the pathogen as well. And there's a concept that's been floating around. It's called herd immunity. You probably have heard of it. That's the idea that enough of the population, if enough of the population is immune to the disease, then the disease spread is stopped, or the disease cannot spread. For COVID, it's estimated to be about 70% or 75%. So if we can actually get immunity or generate immunity in large proportion of the population, then we can reach or achieve herd immunity, and then we can stop the spread. So how do you generate immunity? There are two ways. There's to generate naturally, which means you let people get infected, and then if their immune system gets activated and overcome the infection, then they will be immune. And this actually is happening somewhat. So the new coronavirus has relatives, and they are the common cold coronaviruses. And it turns out some of us that still has a, once we generate immune response, immunity against common cold, we actually have cross-reactivity against the new coronavirus. And this is the reason, at least part of the reason, why younger individuals tend to have less of a disease or symptoms than older folks. And the younger folks tend to have more antibodies against the common coronavirus that can cross-react with the new virus. Another way to generate immunity is through artificial means, i.e. through vaccines or vaccination. So again, borrowing from a slide from this summer's conference, this is Dr. Tony Fauci here talking about the vaccine candidates in the pipeline. So we see these, you probably heard these in the news recently. So Moderna, Pfizer, and AstraZeneca, they're all getting very close to be approved. I think Pfizer was approved in Europe, and it's being decided today, actually, at the FDA. Now, how do vaccines generate immunity? So vaccines basically are pretending to be a real infection. However, vaccines are designed not to cause diseases, right? So vaccine can actually elicit the same immune response as the pathogens can. So B-cell response, once B cells are activated, they can secrete antibodies, and then antibodies can block virus attachment to target cells, so prevent virus from getting into cells. And antibody can also tag the virus for destruction for other immune cells to recognize it, to remove it, to phagocytose it, to kill it. T cells can also respond. So there are two types of T cells. They're T killer cells. They're good at killing infected cells. They're also T helper cells. The T helper cells are actually very important in the immune system. Their job is to secrete cytokines, which are signaling molecules to communicate with other immune cells and non-immune cells in the body. So T helper cells can help T killer cells, can help B cells, can help cells in the innate immune system to get rid of the virus. Now, the T cells and B cells form what's called a memory cells. So these are the cells, once they're clonally expanded, so the clone army, once they're done battling the invaders, the pathogens, then some of them will remain as memory cells. So they're kind of like the veteran soldiers. They're very good at fighting. They know how to fight, and they're very quick in mobilizing. So next time when the real pathogen comes in, then these memory cells will quickly generate clones and quickly fight the invaders. So to protect the body from the pathogens. So that's how it actually works. Now, vaccines can have different platforms. Or another word to say is vaccines have different forms or different ways to make vaccines. There are 3 generations now. So first generation vaccines. So this is the most traditional way of making vaccines. The very first vaccine was made in the 1700s. And they are often what's called attenuated or inactivated whole microbes. So the whole pathogen are either passaged in the laboratory so that they are attenuated or they're weakened, or they're inactivated through chemical means or UV. And we inject these whole microbes into people, and these whole microbes can elicit very good immune response. Second generation vaccines are subunit vaccines, and then they are really not the whole microbe. They're just part of the microbe. Oftentimes, they are the proteins from the pathogens. The third generation consists of recombinant vector vaccine, which are microbes. The scientists can actually engineer microbes to have different genetic makeup. So you take certain genes from the pathogen of interest, and you put these genes into a different microbe. We call them vectors. And the vector or the different microbe can actually become the vaccine once you inject into a person. And then lastly, the newest members of the vaccine platforms are DNA or mRNA vaccines. These are just DNA or mRNA from the pathogen. There's no proteins or any other components. They're injected into the person, and from the DNA or mRNA, viral proteins or pathogen proteins are made within the host cells. And then those proteins will in turn generate immune responses. So immunization can happen with different platforms, can also happen with different schedules. So as you probably have heard, the Moderna or Pfizer vaccines needs 2 doses. So initially, many vaccines will take multiple doses to reach a optimal immune response. And then even after the initial vaccinations done, over the years, there can be booster shots. Like some of you probably had tetanus shots every 10 years or so. And then there's another thing called adjuvant, which are some other substance that mixed in with the vaccine to help elicit better immune response. That can, in the adjuvant, can be all kinds of different components. So let me talk a little bit more in detail about these vaccine platform. So the following 4 slides are borrowed from a Washington Post news article. I think they did a fairly good job of depicting how these vaccine platforms work. So the first one, live attenuated or inactivated vaccines. Go back one. So essentially, you're taking the whole microbes, you inject into the person, and then the microbes, in this case, if it's a virus, then will get into cells and be generated, replicated, and then a cell called antigen-presenting cell will take up the virus, either dead or the live ones, and then break down the some viral proteins and present it to T helper cell. So kind of like hold it to T helper cell. And it also hold it to T killer cell as well. And right here. And then that's how T helper and T killer cells gets activated. B cells will actually bind to the virus particle and ingest it, and then present it to T helper cell, and then T helper cell can help B cell to get activated. So afterwards, T killer cells can kill virus-infected cells, and and B cells can generate antibody, which can block the virus. Several vaccine candidates listed down here, mostly are from China, are using this particular platform. And by the way, some of these vaccines are actually in use, in emergency use in China and some other countries. Next, subunit vaccine. In this case, it's not the virus particles, but viral proteins. They're being injected. And then again, antigen-presenting cells need to take up the viral particles and then present them to T helper cells to activate T helper cells. And they also activate T killer cells and then B cells. And in this particular platform, Novavax, this is one of the vaccine candidate the government is sponsoring as well. It uses that platform and then a number of other vaccine candidates as well. Next is the recombinant vector. In this case, the SARS-CoV-2 gene is inserted into the genetic material of another virus. So AstraZeneca's vaccine uses this strategy. They use a chimpanzee adenovirus as the vector, and then they put the SARS-CoV-2 spike gene into it, and then they inject the vector into the recipient, and then the vector can actually replicate or at least make the antigen, and then again, antigen-presenting cells, T helper cell, T killer cells, and B cells. And then this particular candidate from the Gamaleya Research Institute, this is the one from Russia. I don't know if you've heard. It is being rolled out in Russia, even though the phase 3 clinical trials hasn't really completed. They claim to have about 92% efficacy, but we'll see. Johnson & Johnson is another company that makes a vaccine candidate that's being tested in the phase 3 clinical trial. Actually, it's actually here in Grand Rapids. You can volunteer for that particular trial. And then lastly, DNA or mRNA vaccines. By the way, the previous 3 platforms, we all have approved vaccines for. Whereas DNA or mRNA, we do not have any approved human vaccines. DNA vaccines, we have DNA vaccines approved for animal use, not for human use. mRNA, it's entirely new. There is no prior to COVID, there wasn't any approved mRNA vaccine. So the top 2 or the fastest 2, Moderna and Pfizer vaccines, both use this mRNA platform. In this case, mRNA or DNA is injected into the muscle tissue, and muscle cells or fibroblasts or some other cells in the tissue will take up the DNA or RNA, and then proteins are being made from the RNA, and then from DNA, it first need to make a RNA copy and then make proteins. And then these proteins will be taken up by APCs and then presented to T helper cells, T killer cells, and B cells, and again, generating immune responses. Right. So the mRNA platform has some advantage because as soon as they know the virus sequence, then they can make the vaccine. They just need to decide on which particular protein they want to target, and then they can make a vaccine. So that makes them actually pretty quick in developing a vaccine. Then they can put them into testing. And that's probably why these 2 are the fastest out of all the vaccine candidates. Now, how long does it take for a vaccine to be developed? So this is really, this is a typical one, right? So several of these candidates are actually taking years, right? The average is about 10 years, 10, 11 years, a decade. So we can't wait that long, right? So why does it take this long? So these are the different phases of vaccine development. In the very beginning, there's exploratory stage where scientists are actually doing hard work of studying the pathogen, figure out how they're actually causing disease, and then identify a particular vaccine target so they can make vaccine out of it. Once they decide that, then they need to do some what's called preclinical testing, which means test the vaccine candidate in animals for toxicity and efficacy. And after that, they need to apply for approval, for FDA approval, and that takes actually at least 10 months to get approval. And then they can actually start testing in humans. These are the clinical studies. There's phase 1, phase 2, and phase 3. Phase 1 essentially tests the safety of the vaccine, and then to see if there's any immune response. Phase 2 is often called the efficacy trial, which they'll use a placebo group, compare the group with vaccine, and then the group getting placebo. And phase 3 is a very large-scale trial involving tens of thousands of participants, and then they test further for safety and efficacy. So these takes years to complete. And then lastly, also takes years to complete, is actually large-scale manufacturing. And then the final FDA approval for the whole process. So altogether, it really takes a decade at least to develop. So this I borrowed from AstraZeneca's website. So this is normal, and this is what's happening right now. We shrank 15 years into one. Okay. Really? Why can we do that? Okay, so the reason we're able to do this is because at the exploratory stage, there already being work done on coronavirus. So the very first place where the virus showed up in Wuhan, China, it has a virology institute where they actually have been studying the coronavirus for the last 17 years since the SARS epidemic. So as soon as the virus showed up, they were able to sequence the virus and publish the sequence within a week or two, so that the rest of the world already knows the sequence of the pathogen. And then a vaccine for SARS actually was developed 15 years ago after the SARS pandemic. So we actually have a good idea what would be a good target for vaccine. So they use the same one, the spike protein for the virus. So they now have a target. So within weeks, we already have a vaccine target. So they can actually start making the vaccine and start putting them into test. Several companies, so the mRNA platform, they actually didn't do animal testing. They went to testing humans right away. Why can they do that? Well, mRNA platform has been used in at least clinical trials for cancer therapy, and everybody agrees mRNA platform is relatively safe. Because you're really not putting too much of the pathogen material into the person. So they got approval to go ahead to do animal study as well as human study at the same time. And then FDA, because during pandemic, FDA is giving them green light quite rapidly. So now they are going into clinical study. And then they combine the phase 1 and phase 2 trials together. So they demonstrate it's safe and has efficacy, and then they can move on to phase 3 trials. Now, nobody is actually cutting corners on the phase 3 clinical trials. They know if any bad thing's going to show up, it's going to show up in phase 3 clinical trials. So they enrolled tens of thousands of patients and really did the phase 3 clinical trial the normal way. Now, in a pandemic, there's also helps because in order for the clinical trial to end, you need to reach a certain number of infection cases. And within or during a pandemic, it's actually pretty quick to be able to achieve that. So the phase 3 clinical trial only took several months for them to finish. And that's why we can actually get working vaccines within a year. After saying that, it's actually still very remarkable that we're able to achieve this. Now, one last hurdle, it's still there, is large-scale manufacturing. We don't have the capacity because nobody has ever made an mRNA vaccine before. So they are actually still developing a large-scale manufacturing capacity. They have some good ideas. They still need to ramp up the manufacturing capacity. And that's why the availability of the vaccine is going to be slow for a while. Now, there are concerns. Are we going too fast? This is a survey done in September of this year that's saying many people, 78% of people are thinking we're moving too fast. So they are concerned it's not going to be really safe. However, really based on the data that we have right now on the phase 3 clinical trials involving tens of thousands of participants, it actually showed it's actually pretty safe. The slow process largely is due to financial concerns because the companies will want to have a really promising candidate before they actually move into production phase. Now, with the government help, the government guarantee, the Operation Warp Speed really guarantees the company government money, even if they don't develop a useful or effective vaccine. The government promised them to actually millions of dollars to put into, so they can actually risk the potential for not having a good vaccine and move ahead. So we can actually do that. And then also safety or side effects can be monitored during post-marketing phase, which often called phase 4. If there's any issues, the product will be taken off the market. So now a little bit about the vaccine themselves. I'm going to go pretty quickly. Okay, so the Pfizer vaccine is an mRNA vaccine. It's been shown to be 95% effective and 90% effective against severe COVID. There's minimal side effects. However, there are a couple of allergy cases in the last day or two. When they did the clinical trial, apparently they didn't enroll people who had severe allergies. So they are now recommending people who with severe allergies not to take the vaccine. And so they can produce about 50 million vaccines this year, hopefully, and then about 1.3 billion next year. There are 2 doses required. The thing with Pfizer's vaccine, it needs to be kept at -70. which can be difficult for some field conditions. This vaccine has been approved by the UK government, is almost approved in the United States. The next one is the Moderna one, which is also mRNA vaccine, and it's being shown to be quite effective, about 94%, and it's effective, 100% effective against severe COVID. The participants in the phase 3 clinical trials are pretty diverse. It also requires 2 regimen, but it doesn't need storage at the -70 or ultra cold. It can be just in the freezer. So that actually helps. They are hoping to produce somewhere between 0.5 to 1 billion doses in 2021. The next one is the AstraZeneca one, and this one is a recombinant vector, like I said before. Its efficacy is a little iffy at this moment. Depends on the dosage used. The lower doses actually proven to be actually more effective than the higher doses. So they are actually doing a little more test on it. The benefit of this one is it's significantly cost less in cost. So it will be very good for resource limited settings. And also does not require extreme cold storage. That would help as well. So there are other vaccines. So in Russia, there's this vaccine I mentioned before. They're already using it. And in China, at least 4, actually maybe as many as a dozen vaccines are being developed in China. Some are actually already being used in healthcare workers, and some are already shipped to other locations in the world. Globally, we have actually more than 100 vaccines are being developed, and at least 50 to 60 vaccines are in clinical trials right now. So we definitely have lots in the pipeline. So now we can actually come to this question. Vaccines, are they the answer to a pandemic? Well, how do you really control a pandemic, right? We talked about isolation prevention. So each country actually did some of that. We had to. China did the best job of it. It's basically eliminated the virus, but not everybody can do what China is able to do. And then to reach herd immunity, which is about 70 to 75% of the population being immune. We could let that happen naturally. I think Sweden is doing the experiment there. But with a 2% death rate, it's probably cost too many lives. So we have to do a vaccine. Vaccine, if we can successfully develop a vaccine, then it will actually be an answer. But for many pathogens, we don't actually have vaccine. HIV doesn't have a vaccine. Malaria doesn't have a good vaccine. TB doesn't have a good vaccine. So many pathogens, we don't have a vaccine. But this one looks like we actually may have it, right? It seems like we have several safe and effective vaccines. It's really remarkable we can achieve this within a year. So, do we have an answer? Well, I would argue there's still some questions remaining. We're almost there. Are the vaccines really safe? Yeah, we still have to see. When it starts to roll out, it may actually show up some problem may actually show up. This happened with dengue vaccine, where after they used in the field, they found the vaccine actually caused more severe disease than unvaccinated people. How fast can the vaccine reach the whole population? That's another issue. How fast can they manufacture it? How fast can they reach people? In the United States, maybe less of a problem, but the rest of the world, sometimes it's very difficult to reach places in the world. Now, Another question is, how long will the protection last? We don't know. This virus is so new that we don't know whether reinfection can happen. Actually, there have been cases documented where reinfection happened. You've had COVID. People have had COVID twice. After several months, their immunity actually decreased so that they get the COVID again. So it could happen with the vaccine as well. We don't know yet. And the last question is, will people accept the vaccine? Now, that's another big question. The acceptance rate in the US may not be as great. So Lancet did a survey in May that found 67% people say they would take the vaccine. Males, older adults, Asians, advanced degree holders tend to have a higher acceptance rate, whereas African Americans, Midwest region tend to have lower acceptance rate. And lastly, even when people say they would take it, they may not take it at the end. We can see that with flu uptake or flu shot rates. So another study survey done by Pew Research, they found that from May to September, in May, about 72% of people said they would take the vaccine. And it went down to about 50% in September. I do not want to guess on why, but suffice to say is there may be issues people are concerned with. So many people are concerned about side effects, efficacy, and then some people do not think they need it. Some are concerned about cost. So will people take it? We don't really know. To get to about the herd immunity, we actually need at least 80% of people who are willing to take the vaccines. So are we going to get there? We don't know. I think this is a trust issue. And trust issue is really concerning to us in the society right now. And I would even argue we have a pandemic of distrust going on in our society. The virus is, if you allow me to use this comparison, the virus is misinformation. They're all over the places. There is a mutation out of that, which is disinformation. People intentionally put on wrong information out there. And it's being transmitted through our social media pretty quickly and effectively. And it causes a lot of symptoms: anger, anxiety, frustration, fear, despair, cynicism, hatred, paranoia, contempt. And frankly, I've experienced all of them during this period, not just because of COVID. It's because of all kinds of things: the election, relationship, personal relationship, in the church, etc. So really, we have this pandemic going on. How do we control it? I really don't know. So we can isolate ourselves from social media, or we can try to build up immunity to counter the force of this virus. Maybe we need a vaccine of some sort. Maybe we even need a therapeutic vaccine. So we already have the disease. We can use a vaccine to help our immune system to fight the disease. I really don't know, but I sense a call for us as Christians to really do something about it. The gospel we have is the gospel of truth, reconciliation, of love, and hope. We scientists, we go for the truth, but we know, at least I've experienced it, by just telling other people the truth, it may not be heard. Because if people don't trust you, they will not listen to what you have to say, even if you're speaking the truth. So we really need to practice how to speak truth in love and communicate across the differences. And we are definitely being called as Christians to develop this vaccine, if you will, for our world in this pandemic of distrust. I don't know how the vaccine is going to look like, and then I think we need collectively, we need to really help each other in developing this vaccine. If I may borrow a statement from the Calvin University's Vision 2030, it says, we as Christians were called to seek understanding and promote the welfare of the city and the healing of the world. So I really hope we can move forward in this calling. Thank you. That's the end of my talk.
Thank you so much, Ding. Just a wonderful way to end the presentation, and lots of really valuable information as well, not only about COVID, but about vaccines, the vaccine development. And we've got a lot of time for questions from folks. So again, you can submit your questions two different ways. You can type them out and put them into the chat window, or if you want to unmute your microphone and ask your question orally. We're a small enough group tonight that I think that should work okay. Just maybe as you unmute your microphone, if you could introduce yourself, and then you can ask your question.
So I can end my sharing, right? So that it's a little easier. Okay. So now I can see everybody.
The side effects that occurred in the trials, how hard was it for the medical profession to reduce the severity and keep them from getting worse?
What kind of side effects?
Whatever came up. I hear about pain, difficulty, and that kind of thing.
Yeah, so most of the side effects are tolerable. So they have a particular grade. So they're only 2 to 3% that goes above that grade. And it's mainly headaches or fever or muscle pain. Usually you just take some ibuprofen or some other common medications. And it doesn't actually last long. The side effects last about just a day, and then they disappear. With the severe hypersensitivity, severe allergy, there are EpiPen available, so they can give patient that as well to reverse the allergy. Shall I be checking the chat box?
I can also check that for you if you want to keep. I can keep my eye on it for you. We did have a question from Marcia about the availability of the recording. I know we're recording tonight. Dana, will that be more widely available for sharing?
Yes, we will truncate it, clean it up, and then post it in a YouTube channel. So that link then you can send out to the members after that's done in about a week.
Great, thanks, Dana. We had a question from Dell. Ding, were you surprised about efficacy of 90 to 95% given the small numbers of COVID cases involved?
Not really. I think it's fine. It is small. We less than 100 cases. But clinical trials, especially when the infection rates are lower, it's pretty common. So some HIV trials, they only had a dozen, not quite, maybe 20 some cases. It's actually okay.
It's kind of building on that, I've heard a little bit about some people advocating for trying to do challenge trials. Would that make any, difference in your mind to do that?
Yeah, I wouldn't want to do that. I think we already have enough natural infection going on. It really doesn't morally be acceptable.
Got a question from Bob. Mentioning a study of dialysis patients' blood showing up to 50% of pre-COVID blood samples contain the SARS-CoV-2 reactive T cells. Can you discuss the role of those T cells?
Yeah, so I think that was referring to a study in Europe, in maybe Italy, saying they checked some earlier samples and they found reactive T cells. So it actually could come from two different sources. One is it's what I talked about, cross-reactivity, where these T cells could be responding to common cold. And then now they're actually able to fight somewhat to the new COVID virus. Or it's also possible it was in the fall of 2019 already, although that the chance of that happening is pretty small, unless back then it was a different variant of the COVID-19, which doesn't have such a high transmission rate. By the way, there are multiple studies showing that it's possible during the fall of 2019, it's already in Europe. The virus is already in Europe. Sounds scary. Yeah, I know.
So, quick question: how far back then do the genetics trace this virus? When did it first appear, do we think?
Yeah, that's a sort of $1 million question right now. And then multiple countries try to shed responsibilities. So there's a lot of blaming games. Our president definitely doesn't help in that with that regard. Yeah, so there are some studies showing in the wastewater in Germany back in October, I believe, of last year. And again, if it was true, then it must be a different version of the virus, different variant, that it doesn't have such a high transmission route. So it would just small number of cases and people didn't notice.
I mean, because we're always told, it was a bat in a wet market, and but how long ago was that bat?
Yeah, if you want to ask my opinion, maybe we don't want to put it on the recording. I do have an opinion on that one, which is the bats. Okay, the bats, supposedly the virus coming from, are located in Canton province. And then the wet market is in Wuhan, which is in the Hubei province. And there are like 600 miles between the two. I mean, it's not very easy to move things that way.
Bob had a follow-up asking about, do the T cells confer any immunity? So this is the follow-up on his earlier question.
Yeah. So the T cells, like I said, T cells and B cells. So we often pay attention to antibodies, which are produced by B cells. And T cells are actually very important. T killer cells are important in killing virus-infected cells. Now, why is that important? Because once a virus gets into a cell, the cell becomes a virus manufacturer. So the virus will hijack the cell's capacity of making proteins and RNAs and then use it to produce themselves. One virus gets into a cell, 1,000 viruses can come out within hours. So it's important to kill these cells so that they don't become a virus production unit. And the T cells do that. Yeah.
Ralph asks about your reaction to the natural herd immunity approach in Sweden.
Yeah, I have very mixed reaction towards it. I would say if you have a population that can actually practice social distancing and mask wearing on their own, I think it can work. You don't have to place these lockdown measures in the society. But apparently Sweden was somewhat okay. So the cases right now in Sweden is about 10 times of the neighboring countries. And then they actually recently put into some measures to limit. For example, restaurants cannot sell alcohol after 10 p.m. And they have to do some other things, something like that. A small measure, but they're trying to reduce the spread as well there.
I'm wondering if you could talk a little bit about some of these different types of vaccines. So I have two questions that I was curious about. So one was looking at the Moderna and Pfizer vaccines, both mRNA vaccines, but the Moderna one not needing the really cold temperatures to stabilize it. What was the difference there? I mean, what allows them to be able to keep it stable so much more easily?
Mm-hmm. Yeah, so RNA is actually very unstable. If you take the RNA and you let it get free out of the cell, it will be degraded within minutes. So how can you make it into a vaccine? They have to package it in something. So oftentimes they use a what's called a lipid nanoparticles to surround the RNA in these liposome, basically, a lipid kind of package. So it depends on the composition of this lipid package, then it requires different temperatures. And this is actually how it delivers the mRNA into a cell as well. So the lipid particle will fuse with the cell membrane, and then the content will be delivered inside the cell.
Well, it looks like Marcia's asking what my next question was going to be. Like, if you had a choice of your vaccines, which would you take? So, like, I'm wondering too. Like, once these get distributed throughout the U.S., I'm expecting that there are going to be different vaccines available at different regions, different hospitals. I mean, should we care which one we get?
Yeah. So I would look at their profile and the platform. I don't think there is something inherent to the platform, which one's better, which one's worse. They can't all work. They can't all be effective. They can't all cause side effects or not cause side effects. So with that regard, I would say you just have to look into their particular clinical trial data results to kind of find out what are the side effects. I think all of them is going to be safe in a way that it's not going to make me die. But it can have various side effects. It just depends on. Another thing I mentioned is the adjuvant. They can use different kinds of adjuvant, and oftentimes the adjuvant can cause the side effects. By the way, the side effects are actually the immune system, are signs that our immune system is actually working. So the muscle pain, the fever, and it's actually working, and it's sort of part of the immune response. We hope that we don't have that much of that, but if that's what it takes to generate sustainable immune response, then it has to kind of go that route.
Dell has a really interesting question that he wants your thoughts on. He
Okay.
mentioned that it's not just COVID that's writing on the vaccines. It's confidence in science and medicine leading to the second epidemic you talked about. What are your thoughts on our role as Christians about science, even if it's not specifically our field?
Yeah. So as Christians, like I said in one of the slides, as Christians, we are going after the truth, right? Truth will set us free. And we do our due diligence. We check the references, we check the sources, we do our best to make sure the information that we have is accurate. And then after that, we need to communicate that. And that's the part, like I said, I'm not too sure if your audience don't want to hear, what would you do? So that's the difficult part. Is Rob here? Rob Barrett?
He's still signed on, but I don't know how available his microphone is, maybe.
Okay. So he's part of, I don't know, maybe some of you have heard of the Colossian Forum. I see some nodding heads. Yeah. So they are actually trying to do that. And they have a curriculum. They have a small group format. They're helping people to actually talk across the divide. So that may be, one form of vaccine, but we need more of those, right? So, I don't know how to do that very well. And I'm equally frustrated at times, in despair, and some other times, how to really talk to my friends who are really, my fellow Christians who are in the other camp.
I don't want to put you on the spot, Rob, but I did see the mute icon go away on the square.
Yeah, well, thanks, Hunting. I think that one of the things we're learning from the whole COVID situation is that we have got, as a society, to learn how to work together for social goods, despite our disagreements over a lot of the matters that are involved. And so I think this is a real call to action for all sorts of other things, whether we're learning it from the masks, or whether we're learning it from vaccines, or whether we're learning it about all the other ways misinformation works as a virus, like you talked about. Now is a time for us to learn to be able to work together across difference. And I think we can only do this through practices where we gain the capacity for it. It's not through just learning new concepts. We actually have to take up these practices and build up the muscles that will enable us to do it. And I would hope that the church could be a place that can demonstrate the difference Jesus makes in our ability to love our neighbor in radically powerful ways, especially when our neighbor disagrees with us in fundamental ways, but yet we have the bond of Christ between us.
Thank you, Rob.
And part of this too, we're seeing this as a whole, distrust of science in general kind of movement, in the United States, and well, I don't know how far beyond, but, and part of it is, I think the way science is presented to the general public is science has told us this and science has proven that, it's like, who is this dude Science? I'd like to meet him. He's pretty awesome. Right. I think we need to, people just don't have a great understanding of what science really is. So, it's maybe a bit far to go back when you're just talking to somebody in person, but I always start, my classes with making sure we go through what science is, what it isn't, what it can, show us and, all that good stuff. But I think it goes just back to a complete misframing and misunderstanding of what science actually is.
Yeah.
And misinformation and disinformation as well.
Mm-hmm. Yeah, and I think BioLogos does a good job in communicating that, about science in general.
What part do you think our churches play in this pandemic and the results that are a year, 5 years down the road?
Yeah, I think church is good at doing what church always been doing, caring for the poor, caring for the marginalized, caring for people who cannot speak for themselves. And in this situation, it's the same. And it may be a different group of people. It may be in different contexts or different ways church can do things. And of course, if we're under some lockdown orders, then things have to be done differently. So I still think church is not crippled by the lockdown, whatever measures. We can always have creative ways doing things. It may not be the same ways that we have always been doing it, but it also gives us the opportunity to develop new ways. So this Chinese church I've been part of really used the online platform really well during the pandemic, and there are now more people involved in Bible study groups and prayer meetings than ever before, because they can do it from their home. It's just a lot easier if you have a small child, and you can have the child just playing right next to you. Now you can actually study the Bible or praying with other folks online. So it really can become opportunities for us.
Do you know if the sociology people are looking at how to communicate all this to the African American community? The data you showed shows that they're less enthusiastic about the things that the white community is. How do you communicate that, and how do the sociology people work on that?
Yeah, good question. I'm going to let the sociologists take up this question. Are there any sociologists here?
Claudia, we missed your comment because you guys were muted. Could you unmute and say that again?
I said, I don't think French literature would help very much, but John Calvin might, on the other hand, and he wrote in French.
Yeah, I don't know. I do go to Madison Square Church, and it's a multi-ethnic, multicultural church, and yeah, and then since it's funding, we've been practicing this communicating across cultural differences, and I have to say we're still learning how to do that.
This is changing gears just a little bit, but
Okay.
you alluded to this a couple of times that we don't have vaccines for other diseases that we've been dealing with for many decades. What contributes to that? And I'm guessing it's different for different diseases. Are there some where it's just incredibly difficult to produce an effective vaccine, or is it because of lack of funding? Is it because the disease is impacting parts of the world where we in the U.S. aren't paying attention? I mean, what are the different things that in your mind go into those different situations?
Yeah, those things actually all have a role in it. Maybe not for a particular one. So, for example, HIV, it's not because of lack of funding. HIV has its inherent difficulty. For one, it's a virus that really destroys the immune system. So if you let the virus run its course, it will kill a person 100% time, with very small exceptions. Whereas if you let the COVID run its course, it will not kill people 98% of the time. Right? So HIV, our natural immune system cannot actually overcome it. So that makes that virus very difficult to deal with. So we haven't been able to come up with a vaccine that can outdo the virus itself. So that's that. Malaria and TB, I think largely funding, at least it's a major issue because it's not affecting developed countries. TB and malaria kills more people, a whole lot more people than HIV each year. So various ones. Now, it may be the case that respiratory viruses are easier to make vaccines for. So we have a SARS vaccine, even though we couldn't test it because SARS never came back. Now we have COVID vaccine. We have Ebola vaccine. Ebola is also airborne. We don't have for HIV. We may have one for Zika. We'll see. Not malaria. Malaria and TB. Malaria is a protozoa. It's not a viral. That makes it a little more difficult as well. And TB, it's such a slow growing bacteria. It also makes that difficult as well. So there are various ways. If I want to put it in sort of some kind of context, I would say maybe respiratory viral ones are actually easier to make.
So now that we've sped up the vaccine timeline by tenfold for this vaccine, how does that vow for future vaccines? Will the timeline be sped up slightly in some of those initial steps, perhaps? I mean, I know you have to identify the right protein and all that first, but I mean, is shorten the timeline, or is it just a one-time thing?
Yeah, that's a good question. I hope it would shorten it, but this time, like I said, we owe it to the previous work that has been done the last decade or so, both in terms of vaccine platforms as well as on the coronavirus itself. So we know what the good target would be. And oftentimes for HIV, we still have no idea what's a good target. And then in the future, it really depends on what pathogen we're dealing with. We may or may not be really fast initially. The vaccine platform itself, it definitely has improved in the last 10 years. Partly also due to outbreaks we've had. So Ebola, Zika, and different flu strains that really actually helped us to develop vaccine strategies. Yeah, and also part of the reason why it's slow, like I said, was because of financial concerns. Companies, if you're dealing with a pandemic, yes, everybody's willing to invest. But if you're dealing with something that's TB or malaria, nobody wants to put the whole production in there before they actually have something they think it's going to work. So that would definitely delay the whole process.
So we're approaching 9 p.m. here. It looks like we have time for one more question if anyone has it.
Thank you very much. This was a really great talk.
Thank you. I was kind of nervous. I'm still kind of nervous.
Ding, the teacher came right out of you, I promise.
Oh.
Well, maybe we can all thank Ding. I saw Ralph and Dana both put the applause up, so you can click on the little reactions at the bottom and give Ding a round of applause, or maybe show us on the video here too.
Thank you, guys. Yeah, it's been great. Yeah, great questions.
Well, thanks to all of you for signing on tonight and for joining us. We, as the West Michigan chapter, we are hoping to do another virtual event like this sometime later on in the winter or early part of the spring. And hopefully that can serve as a bridge for us to getting to our first in-person event, which we'd really like to do. So hopefully by late spring, early summer, we might be able to do something like that. But in the meantime, we're going to still try to stay connected together. And if you have topics that you would really like to hear more about, or have other speakers that you would recommend to us, you can feel free to reach out to us by emailing any of the members, me, Ralph, Derek Sherman, Gail Ermer, who's on here as well. And just let us know, and we'd love to be able to see what we could arrange to keep getting together frequently. I also want to mention that Dana posted a link as well, that if there's anyone on here who is not a member of the ASA already, that they have a complimentary 1-year membership available. And so she's got a link there in the chat with the promo code. So we would love to get you involved with the ASA if you're not already. And what a great opportunity to be able to do that for free for that first year. But seeing as it's 9 o'clock and wanting to be respectful of your time, I think everyone can sort of exit now if that is what you would like to do. Some of us will stay on here for a few minutes if you want to chat a little bit more. But again, thank you, Ding, for sharing your time and expertise with us tonight. And thanks to everyone for joining us as well.